So today I'm going to be infecting B cells with KSHV, and I'm going to use the Miltenyi magnetic protocol for this, and we're going to be doing primary tonsil B cells with KSHV. So I have my hood set up, I have my magnet, I have ice, both for my magnet and a larger ice for tubes and stuff. I have the max buffer, which is sterile, the buffer that we use for this isolation procedure. I have my isolation reagents, which come in this little box, and they contain these two little tubes. One of them should be an antibody, the other should be microbeads. I have two filters. I have two columns. I have my pipetters and my tips, and I have a waste container. And I have a couple of Eppendorf tubes here that I'm going to use for cell counting. And I have already had the cells taken out, and they've been recovering for 2 hours in the incubator in 20% FBS RPMI with DNase to keep them from clumping up. So I'm going to get them out of the incubator now. So these two wells are the two tonsil samples that I'm going to be infecting today, 53 and 46. So I'm going to label my two tubes here. And I'm going to label these Eppies as well. And the first thing that I need to do is remove any big clumps of cells that have formed while my cells are recovering. So I'm going to use these cell strainers for that. I just perch it on top of my tube here. Trying not to touch the bottom of it, just to keep it sterile. And then I'm going to use my P1000. I'm going to pipette up and down to make sure that I've resuspended the cells. They could settle onto the bottom of this plate. And then put them into the filter. And then I can get rid of this plate unless somebody else is going to be thawing cells and wants to use it. And now I'm going to use my big pipetter to chase these cells through the cell strainer with 10 mils of max buffer, or 9 mils of max buffer, excuse me, because I want 10 mils total when I'm done with this step. And I can reuse this pipetter only if I don't actually touch it to the filter as I do this. You also want to do this slowly enough that you're making sure that you're not actually overflowing the top of the filter in the process. Since I didn't touch the filter with my pipetter, I can go back into the max. If I did touch the filter with my pipetter, I would have to switch pipettes before moving on to the second sample. So now I can cap these tubes loosely and get rid of these filters. Now I'm going to set up for my count. Cell counting is the next part of this. I need to know how many cells I have in each one of these before I start to actually do the isolation procedure. We use our Moxie for that. And for Moxie, you want to have your total dilution factor to be 1 to 100. So I just put 1 mil into 10 mils. So that's a 1 to 10. So I need to do another 1 to 10 dilution in order to get 1 to 100. So I'm going to put 90 microliters of max buffer in each of these tubes here. And then I'm going to transfer 10 microliters from each one of these to its respective counting tubes. And then we're going to go out into the main lab, and I'm going to pellet the cells while I use the Moxie to count. I capped these tightly, and now I'm inverting them to make sure that I've got a nice even cell suspension. Grab 10 microliters, put it into the correct tube here. Same thing with this, I want to invert it, make sure that it's nice even suspension. Take 10 microliters. And put it in there. So there's my cells for counting, and I'm going to pellet these while I'm counting them. (1500 RPM for 5 minutes). This is our cell counter. This little gray button at the bottom turns it on. I want to load 75 microliters onto the Moxie. This is the little cassette that you use. Each cassette can do 2 samples. This one's been used on half of it, so I'm actually going to grab a new one so I could just use both sides. Or I guess I can just use 2 that have been half used. So it tells you what to do. It says to perform a count, insert cassette into Moxie Z, pipette 75 microliters of sample into the cassette, touch screen anywhere to start. So your cassette goes in, and this is the sample loading port right here by the number. So you want to grab 75 microliters from your tube. Dump it right on. Got to be a little confident because it looks like it's going to overflow, and then hit the circle. And it will suck the sample into the machine and start counting them. So this is 1.2 times 10 to the 6th per mil. So that means that I have 1.2 times 10 to the 7th total, because I have 10 mils at this concentration. And then I did another 10-fold dilution, which gives me 1.2 times 10 to the 8th, taking into account the entire dilution factor. Count the other one. This one is 0.6 times 10 to the 5. 6 times 10 to the 5, which is 0.6 times 10 to the 6. 36 equals 0.6e6. So since I did a 1 to 100 dilution, I can just take both of these numbers and change the 6 to an 8 to get my total. And I don't need these anymore. So while this is finishing its spin, I can do my calculation for what I'm going to do once I go back into the TC room. So I actually don't need that many cells for this. But what I want to do, so I'm probably going to I'll throw away half of 53 so that they're exactly the same. Which means that I'll have 6 times 10 to the 7th total for both of them. And this number is going to dictate how much of the isolation reagents and how much buffer I'm going to add to each one of these tubes for my isolation. So you can see that I have good pellets here. So for this protocol, we want 40 microliters per 1 times 10 to the 7th cells of buffer. And then we're going to add 10 microliters of antibody, also per 10 to the 7th cells. So for 53, I'm going to actually add twice as much because I know I have twice as many cells and I'm just going to throw them away. But 40 microliters times 6 is 240. So I'm going to do 480 and then discard 240. And then for 46, I'm just going to resuspend in 240. And then for both of them, I'm going to add 10 times 6, which is 60 microliters of antibody. So that's what I'm going to do next. I'm going to discard this liquid. I'm going to use my P1000 to resuspend. I'm just going to go up and down and make sure that the cells are resuspended. And then I'm going to use the same pipette tip set at 240 and just throw away half of what's in here. And for this one, I'm going to do 240. And again, up and down a few times to make sure that the cell pellet is resuspended. And then I'm going to take my P200 and do 60 microliters. Of the antibody. So make sure you have the naive B-cell antibody biotin cocktail. Since I went all the way into the liquid, I have to get a new pipette tip. This tube is just about empty. And then I'm going to flick these a few times, make sure it's nice and mixed. And I'm going to keep them on ice for 5 minutes. So this incubation is just about finished, and I'm going to calculate what to do next. So I'm still using this 6 times 10 to the 7th number as my total. In the second step of this isolation, you use 30 microliters of buffer per 1E7 cells and 20 microliters of beads, which in our case, since we have 6 times 10 to the 7th cells in both of our tubes, this is going to be 180 microliters and 120 microliters. So that's what I'm going to do next. I'm going to take 180 microliters of max buffer into each one of these tubes. You'll notice that when I'm using a pipette pen to take buffer out of this large bottle of max buffer, I'm very careful not to get too much of the actual pipette into the bottle itself, because that's how you could contaminate this buffer. So I always tilt and make sure that I'm putting the pipetter into the bottle to the smallest degree possible. I'm going to change this to 120 microliters. I'm going to loosen the cap on the antibiotin microbeads. And I'm going to put 120 microliters of each of these in here. And there is not enough in this tube. So I'm going to have to go out to the lab and open up another kit. All of these Miltenyi kits have the same beads in them. So they are interchangeable. You just have to make sure that you don't mix up the boxes for the antibodies. So I'm going to use the rest of these beads. And just supplement with this new kit. And these are brown because they're magnetic. Flick it a few times so you get a nice even suspension. And then that's going to be a 10-minute incubation. But the next thing I'm going to do is I am going to actually start calibrating my magnetic columns. They have to be pre-washed. So in each one of these packages, there's a magnetic column and there's a plunger. You want to make sure that you keep the plunger nice and clean because we're going to need it later. So I'm just going to open these a little bit. And I'm going to get some 15 mil conical tubes out of my cart. And I'm going to need one of these for each one of my isolations. And then I'm going to need more later, so I'm not going to put them all the way away. Keep your caps nice and clean as well. And once this goes into the magnet, you can actually push the magnet up on this stand so that you have more room underneath for your tubes. And you just want to make sure that the magnet is going to sit nicely inside the tube and not drip all over the place. This is especially important once you start doing your actual isolation so that your cells don't end up in your ice bucket. So I like to do it that way where they're just barely below the edge of the rim, but it's still easy to take the tubes in and out just by tilting the ice bucket. And to get these columns ready for the isolation, what you want to do is just run 3 mils of buffer through each one of them. Max buffer again. Everything in this isolation procedure is max buffer. So I'm taking 6 mils because I have 2 columns. I'm just putting 3 and 3. And those will drip slowly through while the 10-minute incubation is going. And then once these are ready to go onto the columns will be ready for them. It's really important to make sure that you're paying attention to the entire protocol so that you don't waste time, because the longer this takes, the fewer viable B cells you're going to get out to go into your infection. So now that 10-minute incubation is finished, I'm going to get rid of the buffer that I used to get these columns started, but I can reuse these tubes. Label them with the number of my sample. And then I'm going to use my P1000 to apply these magnetically labeled cells to the column. The way this magnetic separation works is that your naive B cells that you want to infect are going to be unlabeled, and they're going to come straight through the column. And everything that you don't want, all of your B cells that are not naive and all of your T cells, are going to stick to the column. And you want to wait until all of the magnetic labeled material has gone into the column, but you don't want to wait too much longer after that to add 3 mils of max buffer to chase those cells through the column. And make sure that everybody who's not sticking, because it's magnetically labeled, actually makes it through into your bottom tube. So I'm waiting until I see all of the liquid go down into the black magnetic resin, and then I'm immediately adding 3 mils of buffer on top. And then I'm going to wait for that to flow through, and I'm going to add another 3 mils. And while I'm waiting for that to happen, I'm going to grab another couple of tubes that I'm going to use to capture all of these bound cells because we want them. In our system, we've discovered that the B cells don't do as well by themselves. They do much better if they have close to the entire immunological milieu that they're used to when they're actually in the experiment. So we save the bound cells and then we add some back after the infected cells are plated. And during that 10-minute incubation, I went to the -80 and I got the virus that I'm going to be using to do the infections after I have these isolated. For virus, today I'm using the wild type BAC16 virus. There are mutant strains, there's KSHV 219, we have several other strains that we use. And this procedure is similar. You just have to know which virus stock you're using and what dose to use it at, which is information that you can get for each virus stock based on when it was made. So for this, to do the second wash through of max buffer for this procedure, I don't actually have to wait until it's all in there. And in fact, this procedure will go slightly faster if I only wait until I basically have enough room to add my second wash. Because this is gravity flow, so the more buffer is in the top, the faster the column will run. And the next thing I'm going to do after this buffer has gone all the way through the column is I'm going to take 10 microliters from each of these and put it into my hemocytometer slide and manually do a count of these cells while I am pelleting them. And then once I know how many cells I have, I can do the math for determining my infection. So these are just about done going through the columns. And what I'm going to do is I'm actually going to do the flow through, which means I take this column off the magnet carefully and put it into a new tube. It's going to have my bound cells. And then I'm going to add another 3 mils of max buffer. And instead of letting it flow through on its own, I'm going to use the plunger to push it through. And that's going to wash all of this magnetic bound stuff through and out. You can see how the brown stuff is coming out into the tube. And then I can throw away this column. So I have my bound cells here. And I have my naive B cells here. I need to count the naive B cells first. I also need to count these, but I don't need to do that until later. I'm going to do the same thing with this one. I'm going to take 10 microliters of my naive B cell isolations and put them onto the hemocytometer. And then I'm going to spin all 4 of these tubes while I'm counting. Again, I'm going to cap and make sure that it's a nice even suspension. I'm going to take these and spin them in the centrifuge just like I did before. So now I have my cells all pelleted and I've done my counts. And there are 7 mils in each one of my B cell tubes. And that's going to be important for the math that I'm going to do. But before I get moving on the infection, I'm going to put these bound cells in the incubator because the infection procedure takes about an hour and I want them to be happy and viable at the end of it. So I'm going to take them and put them in some RPMI media with some FBS and put them in the incubator so that they will be happy an hour from now. I'm going to do 800 microliters of RPMI. And then I'm going to add 200 microliters of FBS so that they have some nutrients to keep them happy. I'm going to label the wells so that I know I'm adding back the correct sample. So I got these counts for my naive B cells, and they can both be multiplied by 7 ml. I'm not quite good enough or confident enough to do that in my head, so I'm going to use a calculator. So that's about 20 million for 46. And about 17 million for 53. And I only am going to infect 2 million cells. So this is going to be a relatively wasteful day for these. If somebody else was in the lab and needed cells, I would offer these to them. So I want to put 1 million cells per 300 microliters in RPMI. Which means I need to do 17 times 0.3 for this one, which is 5.1 mils. And I need to do 20 times 0.3, which is 6 mils for this one. And then we do our infections in these little round bottom flow cytometry tubes. So I need 4 of these, 2 mocks and 2 infected. And I need that many for each tonsil, which means I need 8 total. So this is going to be 46 mock. KSHV. If you're doing a lot of infected cells, this is actually a very small set of infections. If you're doing a lot, I actually highly suggest that you dilute your KSHV in a large amount of media and then split that amongst your tubes so that it's very sure to be uniform. But since I'm only doing 4 tubes, I'm going to pipette the virus into the tubes and then the media into the cells and then transfer cells into all of these tubes instead of splitting them and doing the opposite. So I know that this stock that I have requires 15 microliters per million cells. So I'm going to take my virus and just put 15 microliters in the bottom of each one of these K tubes. This is live virus. You want to be very careful that you don't get it on your gloves while you're doing this. Always recap the vial when you set it down. Always be aware of your pipette tip. Realizing that this is probably in your way. Each one of these virus aliquots, the way we make virus, each aliquot should have about 50 microliters in it. For the most part, you cannot refreeze the virus because it loses too much of its infectivity. So if you have any extra, just throw it away. But don't take out more aliquots than you need in order to conserve virus. So I'm going to throw away the buffer that's on top of these cells. Get rid of these tubes so that they don't confuse me. And then I'm going to look at the calculations that I just did. And sample 46 is going to get 6 mils of RPMI. And I'm going to go up and down to make sure that the cells are resuspended. Notice that I am still keeping the cells cold. The entire infection procedure is done cold. We're going to put them in the incubator after we're done spinning them. For 53, it's 5.1. So now I'm going to transfer 300 microliters because I know now that I have 1 million cells in 300 microliters. And I'm going to do the same thing for this sample. And then for this spin, because these tubes contain virus, we need to be a little bit more careful. Spinning in a centrifuge has the possibility of creating aerosols. So when you're spinning with virus, you always need to take with you the aerosol-resistant lids. So that if something terrible happens in the centrifuge while you're spinning, these lids will prevent the virus from going all over the inside of the centrifuge. So you put it on, and then you move this plastic piece over the top, and you click it. And this spin is 1,000 RPM for 30 minutes. At 4 degrees. So after my spin was done, I just took the tubes and put them into the incubator, and they need to be in the incubator for an additional 30 minutes. And they've now been in the incubator for about 20 minutes, and I am going to get started preparing the plate so that when they're done with their 30-minute incubation, I can plate them right away. So I'm going to be plating mine in a 96 well plate. The math for this is different if you're plating in a 48 well plate, which is actually the more common procedure. But each well of the 96 well plate needs to have 200 microliters total. That's going to be comprised of 500,000 of my infected or mock naive B cells, 500,000 of those add-back cells, which are the bound cells from the column, and then 20% FBS and primacin at 1 to 500 dilution or thereabouts. You can add slightly more primacin if you need to. Primacin is a broad-spectrum antimicrobial. We use it in all of our tonsil lymphocyte cultures because tonsils are inside people's mouths, and the inside of people's mouths is dirty and gross. And if you don't have this in your culture, you will get all kinds of contamination. So to get this mix, I'm going to have 150 microliters from my infections, because remember I did the infections with 1 million cells in 300 microliters. So half of that, 150 microliters, is 500,000 cells. I'm going to add 10 microliters of RPMI and 40 microliters of FBS, because 20% of 200 total is 40. And then I'm going to add half a microliter of primacin to each one of these 50 microliter preparations. So I need to do this bottom line for 16 total wells, because I had 4 tubes with 300 microliters in it for 2 different tonsils. So each one of those 4 tubes is going to be split into 2 wells on the 96 well plate, which is going to be 8, and then 2 tonsils is 16 total. So I'm going to make this up as a mix for all of the wells. I'm going to add this mixture to the wells, and then I'm just going to add in my infected or mock cells on top of that. So 16 times 40 microliters is going to be 640 microliters of FBS. So I'm just going to get a tube to do this mixture in. Move this to the side. And then 16 times 10 microliters of media is going to be 160. And then 16 times half a microliter of primacin is going to be 8 microliters. So then I'm going to add 50 microliters of this to each well on my 96 well plate of feeder cells that I'm going to plate my infections on. So this 96 well plate has irradiated feeder cells in the bottom of it. Primary B cells require feeder cells to stay happy, or as happy as they get. I'm going to take the media out of 16 of these wells. I can keep using this pipette tip as long as I'm making sure that I don't touch it to anything when I'm wasting the media. When you're doing this kind of work, you want to try as much as you can to keep your hands from going over the top of your plate. So I'm holding my pipetter to the side so that I'm not going to accidentally rub against and contaminate the rest of these wells. So now all I have to do is plate my infected cells into these wells. So I'm going to label this plate as I go. Call this ND53, this ND46, and then this column is going to be mock, And that column is going to be KSHV. So I also have to do the 500,000 add back, which I haven't accounted for in what I've done so far. And while the cells were spinning, I counted those cells that I had in the add back wells. So I know that there's 1 ml in each one of these wells where I put the bound cells for my add backs. And I counted them and got 7.6 times 10 to the 7th for this one and 6.8 times 10 to the 7th for this one. And then I did the math so that I know that 500,000 cells is 6.6 microliters for this, and 500,000 cells is 7.4 microliters for this. These cells are already in complete media with FBS, so I don't have to worry about adding back a small volume of these to my wells because I'm not going to be diluting out the FBS or anything like that. So I'm going to use my P1000 to resuspend this well and make sure that it's nice and uniform. And then I'm going to transfer 6.6 microliters from here to each one of the wells for that ND46 tonsil. And then I'm going to do the same thing for 53, only I'm going to add 7.4 microliters. And I'm going to swap these plates out so that I have a minimal distance to travel between them. And just these top 8 wells. Change this to 7.4. Resuspend my other sample. Okay, and now I don't need these anymore. And I've got my wells all ready to receive my cells from my infection. And it is just about exactly the right time. So I grab my tubes. I can just use this centrifuge as a rack, as long as I make sure that I have them ordered in a way that makes sense. Okay. So it's going to be 150 microliters. Then the M's go into the M wells and the K's go into the K wells. And remember, these were spinning. So I need to use my pipette to resuspend before I transfer. And that's it. I can clean up my hood. These can stay in the incubator for however many days that they need to until I do my analysis.